Hello, welcome to a new video class. In this video class, I like to explain the topic in vitro translation. We know that the process of translation which are taking place in both the prokaryote and eukaryote. Translation is the process of producing different kinds of proteins from a mRNA. Usually, that protein synthesis or translation from mRNA takes place within the cytoplasm. But the process of translation in prokaryote and eukaryote differ in several aspects. But when we take the process of transcription, that is the synthesis of RNA, especially messenger RNA from DNA, the location of that transcription vary from prokaryote to eukaryote. Normally, that transcription occurs within the nucleus in case of eukaryotic cell, whereas that transcription takes place within the cytoplasm in case of prokaryotic cell. And one more thing is that in case of prokaryote, both the transcription and the translation are coupled, which means that they occur simultaneously. Whereas in case of eukaryote, transcription first complete within the nucleus, then that transcription product, that is mRNA, move into the cytoplasm. From that cytoplasm, the process of translation starts. So here we are going to study in vitro translation. That is, if the process of translation occurs other than its natural environment or artificially, then such type of translation are called in vitro translation. So let us try to understand what are all the main applications of this artificial or in vitro translation, that is the translation which occurs outside the natural environment, or simply it is an artificial translation. So the main application of translation includes, one is we can able to identify a gene product very rapidly. That is rapid identification of gene product. We can study a gene product very rapidly by using this in vitro translation, that is artificial synthesis of proteins. And like that, we can able to localize the mutation. So localization of mutation through translated gene product, by using artificially translated gene product, we can able to easily localize the region of the mutated region within the proteins. Like that, we can able to study the protein folding. We know that the protein synthesized from mRNA, the protein synthesized from mRNA is a linear chain of amino acid. During the that is called primary protein. During the production of protein, that primary protein undergoes folding and converted into secondary structure, then converted into tertiary structure, and like that. So we can able to study how the protein get folded, what are all the different kinds of bonding which are involved in secondary and tertiary bonding, whether it how the beta pleated structure occurs within the protein, how the alpha helical structure formed within the protein. Like that, we can able to study the whole details. And like that, the fourth one is we can able to insert an unusual or modified amino acid into a protein sequence. Then after that, we can study what happened to the total function of that protein, what happened to the folding of that protein, is there any change that takes place within that protein. Like that, we can able to study. For all these purposes, we can depend artificial translation mechanism. That is the main applications of this in vitro translation or artificial translation. Or we can also say that cell-free translation. We are producing the protein in the absence of a natural cell. So for that purpose, we can depend cell-free extract. for artificial translation. So now let us try to understand how it can achieve, how we can able to achieve an artificial translation product or gene product. So its answer is we can use cell-free extract system for conducting artificial translation. Cell-free extract means we can depend a cell for getting a specific extract in order to perform artificial translation. For example, we can depend E. coli cell-free extract, the cell extract of E. coli, because we know that E. coli is a bacteria, prokaryotic bacteria. Its protoplasm contains different kinds of enzyme needed for the protein synthesis. For example, we know that 70S ribosome, we know that 70S ribosomes are present within the prokaryotic cytoplasm of E. coli. Like that, we need smaller subunit of 70S ribosome, larger subunit of 70S ribosome. We need messenger RNA. We need ribosome releasing factor. We need initiation factor. We need elongation factor. We need termination factor. For all these factors, we need all those factors for protein synthesis. So all those factors contained within the cell. So we can able to isolate that cell-free extract in order to perform in vitro translation. So how we can able to achieve? We can able to achieve in vitro translation by using cell-free extract system. We can get cell-free extract from the E. coli cell, like that wheat grain cell, like that rabbit reticulocyte, and we get insect cell. These are the most commonly used cells for preparing cell-free extract. Now we need to know how the cell-free extract get created. How we can able to make cell-free extract. So first of all, we have to lyse. We have to break down the cell wall components. We have to break down the cell wall and cell membrane of the cell. After that, we have to remove all the unwanted cell debris by centrifugation. After centrifugation, we can get a pure cell extract having ribosome, initiation factor, elongation factor, termination factor, ribosome releasing factor. All after that we get a cell extract containing all the essential factors needed for the artificial translation. That is about cell-free extract. Now I think you will get a clear idea about what is cell-free extract. Now let us move into the main approaches of in vitro translation. There are two main approaches for in vitro translation. One is called standard approach and second one is called coupled approach. In standard approach, we are producing an artificial protein, or we are performing in vitro translation by using mRNA as a template. Whereas in case of coupled approach, we are depending a template, we are depending a DNA template. From that DNA template, first of all, we'll produce a messenger RNA. And from that messenger RNA, we'll produce a protein. So coupled approach means here we are performing both the artificial transcription and in vitro transcription, and in vitro translation and in vitro transcription together. But in case of standard approach, we are using an already prepared messenger RNA for performing in vitro translation. For that purpose, we can depend any criteria or any method. But our aim is to get a template RNA. From that template RNA, we will produce a protein through in vitro translation. But here we are depending a DNA template. From that DNA template, we produce mRNA through in vitro transcription. From that mRNA, we will produce a protein from in vitro translation. That's all. Now let us move into the one more topic related with this in vitro translation. Now, uh, if the protein, if the protein which are going to be produced is very high, or its molecular size is very high, then, and like that, if we need to study the protein interaction within the cell, what are the role of the protein within the cell, where the protein goes and where it binds, what are all the function it mediates. For that purpose, we need to synthesize a protein within the cell instead of producing artificially. So for that purpose, we need to depend a live cell for producing protein in order to study its role inside the cell, what are all the interaction that protein perform within the cell. For that purpose, we can depend different kinds of cell in order to produce protein. So the most commonly used cells are mammalian cells, like rat cell, insect cell, yeast cell, and bacterial cell. Even these mammalian cell, insect cell, and yeast cells are example for eukaryotic cell. Within that cell, protein synthesis and that is transcription, RNA synthesis and protein synthesis take place separately. But in case of bacteria, transcription, RNA synthesis and then protein synthesis are coupled. So here, the main aim of this process, that is recombination protein, we can say that if the protein synthesis takes place within the cell, then such type of protein synthesis are called recombination protein, because that protein, the genes for that proteins are not actually residing within that cell, but it was taken from another cell. For that genes or that RNA, that RNA then inserted into that cell. So that is why we can say that it's a recombination protein. So the production of this recombination protein within this cell need some kind of vector. That all discussed, that topic are discussed in the previous class. But here our aim is, the main application of this technique is that we can able to study the functions of that protein. That is called functional assay. We can also able to create antibodies by using this technique. We can also able to express a protein with a large molecular size by using this cell. But when we depend this cell, we can able to express a protein with a having larger molecular size. So for that purpose, we can depend these live cells. Like that, we can able to study the details of protein interaction. Because when we synthesize a protein in a cell-free extract medium, we cannot able to study how the protein interact with the different cell organelles or a different component inside the cell. But in this case, when we depend a live cell to produce a protein, that newly produced protein surely interact with the different component or cell organ of that cell. Thereby we can able to study its interaction. For that purpose, we can depend a live cell for in vitro protein synthesis, even though it is not an in vitro, because we are depending a live cell. We are producing a protein within the live cell. That's why we can say it is a recombination protein production. Okay. This one also a type of in vitro protein synthesis, even though we are depending a live cell. That's all.